Hi, this is Mr. Young, and in this video podcast, we continue to look at macromolecules. We're now focusing on nucleic acids. In addition to nucleic acid, you should be able to recall our three other major macromolecules, and they are good carbohydrates, lipids, and proteins. And now we tack on nucleic acids. As we consider nucleic acids, I find it incredibly intriguing to consider how at your first day of life, when dad's sperm, as you can see here, we've got a ton of sperm cells, fertilized mom's egg. Here's an egg cell. Only one sperm, as it were, will end up fertilizing and penetrating that egg. The others will not, and they will die off. But how is it that all of you, the information required to make you, is stored in the nuclear contents of a single itty-bitty sperm and this larger egg? In what form does that information come in? Well, there was actually a long period of time where we knew nothing about that. In fact, a lot of smart money was on proteins being the genetic material that was responsible for carrying information that would be passed on from parent to offspring. But when it was determined, in fact, that DNA was the genetic material, then the race was to figure out what did it look like. And as we look at nucleic acids today, that's going to be a major focus for us. How does the structure of DNA relate to its function or its job? Well, in the early 1950s, the structure of DNA was a big mystery among the scientific community, and there was a lot of competing scientists trying to determine what the structure was. The picture you have here represents one of the pictures taken by Rosalind Franklin, and in this diagram that you can see, it was gathered by taking essentially crystallized DNA and placing it under X-ray and gathering the images. Well, this is one of the images that Rosalind Franklin took. And as you look at it, you try to deduce three-dimensional structure from it. That can be really difficult. But to the trained eye of Francis Crick and James Watson, it was readily identified to be none other than a double helix. And that is a phrase that's often used to describe one type of nucleic acid, and that being DNA. A double helix. Now, we've seen the term helix before as we saw alpha helices in protein structures. The helix term just means this sort of spiral shape that you see here, or a twisted ladder, a spiral staircase. All of these used to describe the molecule of DNA, one of the nucleic acids that we'll talk about today, and the genetic material. And so as we continue to look at the structure of DNA, what we'll find is that this double helix, which is noted by the deoxyribonucleic acid, there's where the DNA comes from. This double helix is comprised of a lot of small pieces, just as we've seen before with proteins being comprised of monomer units of amino acids and carbohydrates being comprised of monomers like monosaccharides. Here we see DNA is likewise comprised of smaller subunits. In these cases we refer to them as nucleotides. So nucleotides are the building blocks that make up the larger molecule of, in this case, DNA. In fact, if we put a bunch of nucleotides together, then we'll just add the root poly, meaning many nucleotides, or polynucleotide. Every nucleotide, you need to understand, has three parts that make it up. In the case here, you see two parts make up the backbone of this molecule, and those two parts here are shown to be a sugar unit that we'll describe later in more detail, and a phosphate group. And then one part that's used to make up essentially the rungs of the ladder, so to speak, in this case, nitrogenous bases. And that's what all of these are referring to down here. These are nitrogen-containing bases. And in your studies before, perhaps you've seen them represented as A's and T's and C's and G's. But what we find is that in the human genome, for example, we have 3 billion base pairs, or in other words, pairs of these nucleotides that join together. Now what we want to identify is what you can see here. Not every nucleotide joins with every other nucleotide. There are specific pairings. And here you can identify how C's bind with G's, or cytosines with guanines, and thymines with adenines. And that pattern will be repeated throughout all of the molecule. And so the base pair rules are rules that you just have to know. For every A, there will be a T. For every C, there will be a G. And there's been research done to support that, where we found that the ratio of adenines to thymines is about 1:1. The ratio of cytosines to guanine is likewise 1:1. And thus we see support. Look back at Chargaff's rules when you later do your studies on the structure of DNA. As we continue to look at this structure of DNA, we'll break those nucleotides down in a little bit more detail right now. The 4 nucleotides for DNA, again, these are all nucleotides, are shown here. Now, what do they all have in common? That's right, they all have this green blob, whatever this green blob is here. It's the same in all. Now, if you were to break it down into its different component atoms, you see that we've got a lot of carbons. In fact, 1, 2, 3, 4, 5 carbons. Not only that, but we've got a lot of hydrogens. Remember, this carbon will have a single hydrogen sticking off it. This carbon will have 2 hydrogens sticking off of it. So we've got a lot of hydrogens. We also have a lot of oxygens, and they seem to be in this ratio of CnH to the 2NO to the N. What is this looking more and more like? That's right, this is none other than a sugar. And so our first component of a nucleotide is a sugar unit. And those sugars, as we find out, happen to be a pentose sugar. Pentose, referring to the fact that it is a 5-carbon sugar. Notice the ring structure as well. That pentose sugar will vary depending upon the nucleic acid we're talking about. In the case of DNA, we'll call it deoxyribose. And in the case of RNA, we call it a ribose sugar. And so for DNA, it is deoxyribonucleic acid. For RNA, it is ribonucleic acid. And that's, again, that D or that R is just related to the sugar unit. So they all have the same sugar unit, but they all also have this same structure here. We refer to this as a phosphate group. So we've got the sugar unit as the first common feature. The second feature of all nucleotides is a phosphate group. And that phosphate group is shown here to have a central phosphorus atom. Then you have 4 oxygens coming off of it. In fact, in your chemistry work before, you may have remembered we've got this polyatomic ion of phosphate with a -3 charge coming off it. This is a phosphorus atom, 4 oxygens. There you see them right there. And so this phosphate group with that negative charge is what results in this negatively charged backbone. Linus Pauling, in fact, in his predictions of the structure of DNA, had actually drawn a triple helix, but the thought that you could have these 3 helices with negatively charged backbones in at one another in this helix, that was impossible. Only a double helix would be possible. Well, in addition to the phosphate group, then, now we have the unique parts about each nucleotide. That's shown here in these nitrogen-containing bases shown on each of these nucleotides. So for adenine, again, this is adenine, represented with an A, we see a 2-ring structure. For guanine, we also see a 2-ring structure. And in fact, the similarities that we see here with those two are what result in them being grouped together in a group of nucleotides that we refer to as purines. Purines, or nucleotides that are characterized by having a 2-ringed nitrogenous base, that being adenine and guanine. The other nucleotides that you can see here, that would be thymine, cytosine, or even uracil, which is another base added in RNA. These are all considered to be nucleotides that we refer to as pyrimidines. And so one of the things you're expected to be able to do is to differentiate between purines and pyrimidines. Again, the difference being a single-ring nitrogen-containing base for pyrimidines versus a double-ring nitrogen-containing base for the purine nucleotides of adenine. Well, what do those nucleotides do in order to form the double helix? This is a great diagram that really simplifies it nicely to show you the sugar and phosphate backbone that you can see here, followed up with the nucleotide bases, or the nitrogen-containing bases, that are connected together in the middle by hydrogen bonds. Hydrogen bonds are relatively weak bonds, and those hydrogen bonds allow for a connection between one strand of your double helix. In other words, when you are to think about this double helix like so, this ribbon itself that we've drawn is represented by the sugar and phosphates shown here. Those are negatively charged backbones. But then in the middle, we have bases that connect to one another. Bases that connect to one another like so. And those connecting bases are held together in the middle by hydrogen bonds. Remember what we talked about earlier, our base pair rules. C's with G's, C's with G's, A's with T's. Notice again the difference in the hydrogen bonding number. Here we see with our cytosines and guanines 3 hydrogen bonds. With our adenines, our thymines, only 2 hydrogen bonds, so a little difference with that as well. Finally, you can see how we have somewhat of an upside down strand on the left side versus the strand on the right side. They are what we refer to as anti-parallel. They're not going in the exact same direction, although they are parallel to one another. And so we use the word anti to describe simply that they are in opposite directions, and the term parallel to describe the fact that they are in fact right next to one another, going in parallel fashion through this double helix. So what do nucleic acids do? Well, they are simply the recipe for proteins. And by they, I'm referring to what we normally think of when we think about nucleic acids, that being DNA. DNA is your blueprint for life, and it is a recipe for proteins. But what good is a recipe if it just stays in the recipe box, right? Likewise, what good is your DNA if it just stays and its information just stays within its container where it's stored, and that is the nucleus. Now, granted, yes, we do have DNA found outside the nucleus, extranuclear DNA, will be found in 2 other organelles. Do you remember what they are? In the case of plants, chloroplasts, absolutely, that plastin. And in addition to that, we see mitochondria have their own DNA. But here we're just talking about nuclear DNA. And in the case of the nucleus contains DNA, and the DNA is locked up in there. It can't get out. And we don't want it to get out, frankly, because do we ever want the recipe to get dinged up outside where we have DNAases, proteins that will degrade the DNA? No, we got to keep it safe. But we want its information to get out. And we want it to get out because here's why. If DNA is a recipe for making proteins, and the recipe is stuck in the nucleus, shown here, but proteins are made up in the cytoplasm, then we have an obstacle to overcome. How do we get the information from the DNA into the cytoplasm where proteins can be made? It has everything to do with the intermediate. That shown here is RNA, or ribonucleic acid. What's one difference you can already tell just by this image? Good. DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule. In addition, DNA has those bases of A, C, T, and G, whereas RNA has A's, C's, G's, and U's. No thymine, only a molecule called uracil that acts as that 4th nitrogen-containing base. And finally, RNA happens to be mobile. There's an M that will tack on in front of this molecule, RNA. As we see, it's a messenger. And it will be used to take information from the DNA, carry it out to the cytoplasm, to be read by a ribosome. And so in the process of making proteins, or protein synthesis, that you'll learn about later as you study the central dogma of molecular biology, we'll see how DNA is the recipe, but its goal is not to just stay tucked in its little recipe box. Its goal is to be expressed and to be made into whatever the cell is needing. And that product, in the end, we see to be proteins. And so this is a good simplified version of how nucleic acids work in your body and mind, and I hope that this helps.